Today, I'm going to show you an application that we developed for analyzing RNA sequencing data. My name is Stephen G. from South Dakota State University. The website is here, and it is designed for analyzing gene-level expression matrix that was derived from RNA sequencing or DNA microarray experiments. It is a graphical user interface website, but it's powered by a large number of R and bioconductor packages. We also have annotation databases like annotation of genes and pathways that is integrated into the database. We also offer API access to a few web servers like StringDB. And it was designed for busy biologists, and so it's very easy to use and it's very graphical. If you look at the design framework, so we have, once your expression matrix is uploaded, so again, so this is not raw RNA sequencing data, it's the summarized gene level expression data. So we go through a pipeline. This pipeline, each step uses some R packages or some special or some R functions to clean up the data, visualize the data, exploratory data analysis, identify differential expressive genes, and then we're going to conduct pathway analysis using a variety of packages. And enrichment analysis or gene set enrichment analysis in here. And there's a lot of databases supporting this application. So with this, once you upload this data, it very quickly can get a lot of plots. So these plots, for these, right now, these are diagnostic plots. And you can also get exploratory data analysis, different plots, and these plots, heat maps, T-set maps of the genes, and Kmeans clustering, enrichment analysis based on Kmeans clustering, and principal component analysis. Okay, we even have transcription factor binding motif enrichment analysis based on Kmeans clustering, and then differential gene expression. For differential gene expression, we have DSIC2 packages and also Lima package. We can analyze, identify these differential expressed genes and also visualize their overlaps using Venn diagrams, and visualize the protein action networks using the StringDB API. And we can show different plots of differential expression. Some of them are interactive. We can conduct pathway analysis on these genes that was up or down regulated using different databases, and we can visualize the enriched pathways by using a tree like a map. For pathway analysis, we have 4 or 5 different packages that we're using to analyze this data in terms of pathways and different visualization. One of the most important things is visualizing the gene expression on KEGG diagram. So the KEGG diagram was fetched from the KEGG server, and then gene expression data was overlaid on top of it. And we can also visualize gene expression on the genome. We can identify regions of the genome that was enriched with genes that was up or down regulated. So these are all the different plots that you can get in a very short amount of time once you upload it to the website. Let me show you how this website works. Before I do that, so this is the input file, how the input file looks like. So you have the first column has to be gene IDs and then followed by gene expression data. The most we recommend to use we recommend to use RNA sequencing read-const data if that's available, because with read-const data we can use DSeq2 and use the negative binomial distribution to identify gene expression, differentially expressed genes. Another thing I want to mention is once you have the gene expression data, and we'll name the columns carefully so that the groups are naturally defined. So the replicates was clearly labeled by _1, 2, 3. So this is control samples. These are the experiment samples where we have HOX-A1 gene knocked down. Okay, so iDEP will read these column names and trying to divide samples into different groups. The gene IDs here is ensemble gene IDs, which was used internally, but you don't have to use ensemble gene IDs. You can use other gene IDs like gene symbol. You can use gene symbol, gene IDs, and you can have multiple groups. Here we have 4 groups. We have wild type and P53 knockout samples, and they are radiation treatment or mock treatment. So we have 4 groups here. Another type of data you can upload is RNA sequencing FPKM numbers. So here's FPKM numbers. The gene IDs are gene symbols, which will be automatically converted into ensemble gene ID. And lastly, the one type of data is a very special type of data is that you can have gene IDs followed by fold change and FDR values. For example, this could be something that you got from a CUF-DIF analysis. And one thing you want to do is you want to put them together in one file. So different comparisons, fold change, log2 fold change, log2 fold change, and corrected p-values in different columns. So you can use, you can analyze this type of data. So if you go to this website, by the way, this website has been used by over 1,800 people from all over the world, about 62 countries all over the world. So some people come here frequently, and right now there are 3 users using it, as you could see, from different protocols. Okay. Okay. So let me show you how I use this website to analyze gene expression. This is RNA sequencing read-const data. This is normalized gene expression data like FPKM. This is the fold change data that I showed you about. So today I'm going to show you a very simple data, which is the human cell line was treated, was knocked down with HOX-A1. So I have 3 control and 3 experiment samples. It's a very simple experiment design. If you have a very complex sample experiment design, and one of the things you could do is you can just partition it into separate files and upload to here. Obviously, we have better ways to do that. But if you're really confused, and you can, that's an option. So once this file is uploaded, as you could see, the iDEP software trying to look at the gene IDs, trying to make a good guess on what type of species is that we're dealing with. Here, it guessed that it's probably a human sample, which is the correct. So sometimes it doesn't guess the correct ID, or it will show ID not recognized. Sometimes you can also choose this new species and upload a pathway data to iDEP to analyze your own data. So I have some example data on what type of data files. So if you click on the question mark on each of the page, that will lead you to the documentation side on WordPress. Okay, so this explains the details about the data formatting, and also gives you example files that you can use. And also one other thing that I want to show you here is there's a few smaller tools that we developed to help you prepare your file. So one of this is this tool here. This tool here what it does is will extract a read count FPKM value from multiple files that was produced by something like this RNA sequencing read count summarization pipelines. You upload a group of files and you specify which columns you want to extract, and this will combine these files. Okay, so we have a few other files. Another tool that is very useful is here. So this tool is to convert gene ontology files. So if you have a file that for each gene you have a gene ontology file, this will convert it into a GMT file. Another tool that is here, I think this will be very useful, is that here on this tool you can download a lot of gene expression data that was publicly available and also processed. So there was about 3,000 human RNA sequencing data sets and about 3,000 mouse RNA sequencing data was processed by these people here on this website. Okay, you can download that. So these are the tools and documentations. So let me show you how this website works. So once you upload this file, you can go and check into each of these tabs for your result. So the first few tabs: pre-process, heatmap, Kmeans, and principal component analysis. So these tabs are for exploratory data analysis. And then we have differential gene expression and pathway analysis, and visualizing genes on the genome, and bi-clustering and co-expression network analysis here. So once you click on these tabs, immediately analysis was done using the default settings. So here we're showing the total number of read counts per sample. So we have some samples, for example, the control number 3 have about 20 million read, while control number 1 have less than 14 million. So there's a little bit of difference, but not too much to worry about. These are important because some of the analysis we need to know if there's a big variation in sequencing depth. Okay, so for details, read the documentation side. One of the things that I want to let you know that some of the plots are responsive to your browser. So if you make it narrower, as you can see, this plot becomes narrow, and if you make it really narrow, will be something like this. But not all of the plots. So these plots are not. So this is the on the left side of the panel, you have things that you can do to customize your analysis. For example, the filtering. So by default, genes that are expressed lower than 0.5 counts per million in all of the samples was removed. So the counts per million was calculated like this. For example, here for a 20 million library, a 0.5 counts per million will translate into 10 counts. So if a gene is not expressed in at least 10 counts, and one of the samples will be removed. Okay, so obviously you can change this. So for read counts data, we need to transform it for exploratory data analysis. So there are a few options that you can use to choose what type of options you want to you want to use for transforming. The default is just a log transformation by adding a constant. So all the consoles added a 4 and then take a log 2. So that's the default. If you want to know the details, you can go to this website, go to the documentation site, and on the pre-processing, for example, it shows the effect of different type of transformation. The VST is very aggressive, while the RLog is less aggressive, but it's kind of slow. Okay, so what you want is that this is between technical replicas. You don't want to see this type of variation at the lower end of the spectrum for the reads for genes that are detected on the lower end of the spectrum. So you need something like this, but you don't want to be too aggressive. Okay, so you can use this scatter plot at the to guide you. For example, if you want to be further reduce the variability in here, you can choose to increase these constants to 8, for example. If you change the default, everything will be recalculated. And if you go down here to see, as you can see, this it is a little bit of reduced the variability. But we have tried a lot of we tried very carefully to change this default setting so it works very well. Also, this count also depends on total number of reads that you have in the library, and this distribution here shows you the transformer data, how it's distributed. So we don't want to see a lot of differences between the replicas, and also we don't want to be dominated by the lower end of the counts. Okay, so this is the box plot. One other thing that you can do here is you can plot gene expression for a selected gene or group of genes sharing the same name. So for example, this is HOXA1 gene, which was knocked down in this experiment. So alongside HOXA1, it also shows HOXA2, 3, 4, 5, 6, 7, and 10. Okay, as you can see, HOXA1 gene was downregulated in the knocked down sample, but so is HOXA5. HOXA5 is also dramatically reduced, and HOXA2, but not HOXA7. Okay, if you want just one gene, you can see HOXA1 and about HOXA10, so that you can add a space character, and then we only have one character, and you can see the line graph for each individual graphics for each individual samples. Okay, the control samples, the knockdown samples. Okay, so since these scores are log-based too, so you don't see a huge difference. Actually, there's more than as you can see, there's more than 2-fold reduction in the HOXA1 gene expression. Okay, so you can also download the processed data and the converted data. So by converted const data, I mean if your data is not using the ensemble gene ID, so we're going to convert it into ensemble gene ID. So if multiple genes are mapped to the same ensemble gene ID, we're going to keep just one of the genes with the highest variation ranked by the standard deviation. So in the Heatmap tab, what we are presented with the heat map of the data, which shows you a very give you some idea of how the data looks like, the whole holistic view of the data. Again, this is very important to get an idea of the general gene expression pattern in your data before you're conducting a differential gene expression analysis. A lot of biologists are not spending enough time in exploratory data analysis. They go strictly to business. So again, this is like skipping the dating part and go directly to getting married and having babies. So skipping the fun part of it, and it's also very important part of the process that you get to know the data, and which is very important for the decisions you make down the road. Right? Sometimes you might find that your data is actually have too much noise, or have something wrong with it. Okay. So for example, you might find that technical replicas are actually mislabeled or something. So the big things that is important again. All right, so in this page you can customize a lot of part about this gene expression data. You can customize the color. You can customize how the genes, how the data is processed. For example, you want gene to be normalized by gene, so that means, as you can see, the figure looks very different, because what we are doing here is, in addition to subtracting each gene by the median, the mean, and we are also divided each row by the standard deviation, so every row have the same standard deviation. Okay, so another thing that we have here is we have an interactive version of the heat map, which you can zoom in and zoom out, and if you mouse over, you can see the gene symbol. Okay, and you can see, okay, I'm going to need 1,700 genes here. So for this analysis now, all of a sudden the data looks like this pattern. You can zoom in by clicking and dragging up and down. For example, if you're interested in these genes, you can zoom in and to see what genes they are. Okay, you can zoom out by clicking here, and then you can zoom in to a small portion of these genes. For example, if you're interested in these genes that are very different within between technical replicas, you can zoom in and take a look at what type of genes. You can also save this as a graphics. Okay, you can save that as a graph. And also, this plot is responsible to your window size. If you resize your window, this graph will look different. Please. Okay, this graphics is, well, okay, so if you zoom out and if you zoom in, again, you'll show you can also see the correlation matrix, how things are, the correlation coefficients were calculated by top 25% genes ranked by the mean gene expression. And you can see the control samples are highly correlated, and the experiment sample that with the OCT1 knockdown was highly correlated, and you can see their expression correlation coefficients, or you can skip them. You can download the high resolution. You can download the data behind it if you like. Okay, so, and you can have a hierarchical clustering tree of this. Okay, so this is the same thing for this heat map. You can download a high resolution map for a high resolution figure for publication, which is this is requested by some user. As you can see, these samples and these graphics, you can zoom in and see the graphics. It looks very good. If you don't want the tree to be for the sample, you can disable it. You can disable the tree, and so your sample will be the same order of whatever you produced. Okay, so in the Kmeans tab, what we are doing now is we're running a Kmeans clustering on the top 2,000 genes with the most variation across samples. So we rank the genes by standard deviation, and we take top 2,000 of the genes. The same thing was for the Heatmap, and I conducted a Kmeans clustering. And so we divided these 2,000 genes into 4 clusters. Okay, so the 4 clusters are clearly labeled here: cluster A, cluster B, cluster C, and D. They have different type of genes. For each cluster of genes, we automatically conduct enrichment analysis. For example, cluster A genes are enriched with cell cycle related genes. Cluster B is cell cycle as well. C is cell surface receptor signaling pathway, and D was nervous system and other things. If you're the 4 clusters are default. In this case, it's not very reasonable. As you can see, A and B are very similar. But how do we decide how many clusters we use? Is that you can use this diagnostic plot. As you can see, the 1 cluster, 2 cluster, we have a huge reduction in the within group sum of squares. Okay, but if you increase that from 2 clusters to 3 clusters, there isn't significant decrease. So in this case, we decide 2 clusters is actually the best solution. So we go ahead and reduce it into 2. So 2 clusters. Obviously, this is a very simple experiment, and there's a very little variation within the groups. So 2 cluster actually, we have genes upregulated in the knockdown and genes downregulated in the knockdown. All right. So another decision you have to make is top 2,000 genes. Why top 2,000? Why not top 1,000, top 3,000? So there's this plot will help you make that decision. So this is plots the distribution of genes by their standard deviation. As you could see, by top 2,000, you are getting all of these genes. Okay, if you want top 1,000 genes, you can reduce it to here to top 1,000. Okay, and you have 1,000 genes that was divided into 2 groups of samples. Okay, unfortunately, it's running quite a few analyses, but that's fine. Our server is very powerful, so feel free to play all different type of settings. And once it's done, we'll repopulate everything. Okay. So for example, cluster A have 460 genes, cluster B have 540 genes. Okay. So the gene, so with top 1,000 genes, we're getting, the distribution like this. There's something wrong with this code, so I'll fix that later on. So here, in addition to the enrichment analysis, which by default is a gene ontology biological process, we can also use KEGG enrichment analysis. For example, here cluster B, which is this cluster was downregulated, was enriched in cell cycle related genes. Cluster A very significantly, 10 to the negative 14th. And cluster A, we have cytokine receptor interaction, some kind of immune response. Okay, genes. We can also change it to a lot of other pathway databases we have. This is human data. We have StringDB database that we downloaded from the Broad Institute. And for example, one thing we can do is we can see what transcription factor transcription factor binding motif. Yes, motif. Transcription factor binding motif was enriched in the genes. For example, E2F factor binding motif was enriched in the target genes, was enriched in the downregulated genes. Obviously, we know the E2F factors, including E2F1, was a major regulator of cell cycle genes, which makes sense. And the RNA-Seq and also a lot of other databases that we collected enables you to ask a lot of other questions, okay, including mRNA target genes and cancer-related genes location, oncogenes. Okay, so for the human data set, if you're working on human, and we have a large set of in addition to RNA-Seq, if you come to a website that lists the sources, so we have Ensemble, gene ontology from Ensemble, we have RNA-Seq, and we also have GeneSetDB from, and we have a lot of other transcription factor target gene database. Okay, we have mRNA target gene database. In total, we have 72,000 gene sets available in here. So you can ask a lot of other questions. One other thing before I move on is this. We also for iDEP, we also downloaded the transcription, the promoter sequences, and scanned the promoter sequences with transcription factor binding motifs. So these, and then we store the scores and we run for each gene cluster we identified from Kmeans clustering. We also run enrichment analysis of the binding motifs. For example, we also identified E2F family binding motif that's enriched in cluster B, which is very significant, and also some other transcription factor families. Okay, so if we switch back to gene ontology, okay, as we know that gene ontology and a lot of other websites, so sometimes there's a lot of overlapping between different terms for cell cycle, and then here cell cycle process. And these terms are very related. So to solve this problem, we need a way to visualize the redundancy and the overlap of these pathways. So our solution is this. So we have a tool that we developed, and we put these terms into a tree. So for example, the cell cycle and cell cycle process, their significance was visualized by this dot, the size of the dot. They're highly overlap, as well as these guys, they're overlap. So all of these guys are related to cell cycle. Okay, so this is a good tool that that is available here. Again, you can download the high resolution map, which you can zoom in or submit for publication. Okay. The enrichment result, this result, you can also be downloaded. You can download this map. Not only you have the p-values, also you have the genes that is related to each one of the samples, each one of the terms. Okay. So is the Kmeans clustering data. You can download the Kmeans clustering data, and then you can see each gene, and you also the cluster which cluster it belongs to, and the gene expression data. So in case you want to analyze or realize data in a different format. All right. So this is the Kmeans clustering, and next is the principal component analysis. This is very simple. So what we have now is mapping of the samples by principal component 1 and principal component 2, and the sample types was labeled by data point. Clearly, in this case, principal component 1 explains 82% of the variance, and principal component 2 explains 6% of the variance. So the first principal components captures a vast majority of the variants. So, and then we see a clear difference between these two classes of the control sample and the knockdown sample. Within the knockdown sample, there's some variance was captured by principal component 2 or 1. Okay, so similar to principal component analysis, we have multi-dimensional scaling, and we also have T-SNE visualization. Okay. One of the things with T-SNE plots is dependent on random numbers, so you can repopulate this random numbers and to get slightly different plots. Okay. Another thing that, well, obviously we can download the high resolution figures from here, and we'll also download the coordinates of all these plots. Okay. Another thing that's a little bit novel is that we are running pathway analysis using the principal components. Okay, so principal components. So for each of the principal components, you have a loading projections on each of the genes. Okay, so we take this number, and then we treated these numbers of the loadings such as similar to a fold change or gene expression value. Okay, and then we use that for to run pathway analysis using a program called PGESA. Okay, so as it's recalculating now, as you could see, what we got is the correlation between each of the principal components and the pathways that we have here. So we have cell cycle genes. Cell cycle. This is cell cycle related genes that are significantly different between different principal components. For example, the first principal components was negatively related to the cell cycle related pathways. Okay, to cell cycle really, and the response to growth factor and cellular response to growth factor are higher activated in first components. And this is if you have questions about how to interpret this, you can read the documentation on this page. So this established a relationship between the components, principal components, and the pathways. Okay, so the next step is differential gene expression. As we said, iDEP look at the column names and defined groups in these samples and conducts pathway analysis between the two groups of genes. Okay, by default, it's identified 791 upregulated genes, 1,176 downregulated genes realized in this chart. And also, in this case, it's very simple. We don't need a Venn diagram, but we can have a Venn diagram. Out of the 11,000 genes that pass the filter, we have 2,147 genes that are downregulated genes, and you can split it into upregulated, downregulated genes. Okay, so you can download the gene list. You can also download the gene list and the sample. If you have a more complex experiment design, you can use this window to define your comparisons, and you can recalculate your differential gene expression numbers. You can also upload an experiment design file and use this to construct very complicated statistics models. All right, so once you have these differential expressed genes defined, you can examine each one of the gene list of differential expressed genes on the DEG2 tab. So here we are showing the differential expressed genes of each of the comparisons. In this case, we only have one comparison, so by default it shows the expression data, and it also shows the volcano plot of lactophotons and the FDR here, and you can also switch it to a interactive plot in here, where you can mouse over, you can zoom in, and you can see what gene it is. Okay, the same thing goes for MA plot. It's the same thing. So you can see what genes is it. For example, this gene was very different. This AREG gene is also highly upregulated. Okay, and same thing for scatter plot. These are a little bit. This is static plot, and then this is interactive version. We can also, similar to a Kmeans clustering, we can also identify transcription factor bonding motifs in the promoters of up or down-regulated genes. Okay, so what we are doing here is in down-regulated genes, the most significantly enriched bonding motif is the E2F bonding motif. And up-regulated genes with SP2, but is not as significant. As you could see over and over again in this application, you got a lot of results, but don't get too excited by each one of those. Try different method, try different combination to see what is a really robust result. Okay, you can similarly you can have enrichment analysis of the up and down regulated genes. This is very similar to Kmeans analysis, but in the Kmeans analysis we are only using clustering analysis. But here the difference is we are dealing with up and down regulated genes, and for each of the gene list we are doing enrichment analysis. Similar, we have a lot of options. So we can, have, for example, mRNA, microRNA target genes. Okay, so the down-regulated genes we have MRI 192. So these type of microRNA target genes are enriched in the gene list. So I think we have a transcription factor target gene. Transcription factor target genes in here. So the down-regulated gene, we have SP2 target gene, we have E2F target genes. That is defined the TF target genes. Okay. In the up-regulated gene, we have an F-coupled-B target gene that we enriched, and that is, based on. So the up-regulated genes was immune-related genes and cytokines. Probably an F-coupled-B plays a role. As you could see, using the iDEP enables you to really generating in-depth molecular hypothesis about this gene that you are dealing with. Similarly, you can have a tree structure that realize the overlapping terms or pathways, and this is probably better with the high-resolution graphics in here. Okay, in this high-resolution graphics, as you can see, these are really so the E2F target genes was down-regulated and is very significant. For example, E2F1 target gene and E2F2 target gene are significant. The E2F4 is somewhat overlapping, but it is two different branches. Okay, another group is AP1 and TAD1 target genes. Okay, For the upregulated genes is F, copper B, RELA, gene SP1, and this seems to be making sense from what I know about cell cycle. And because we downregulated, we injected some microRNAs into the cell lines. It probably triggers the cell cycle arrest, and also in the same time triggers the immune response of the cells because exodus siRNAs was injected. All right, so okay, so you can obviously spend a lot of time examining of this. One of the really cool things that we added very recently was the API access to StringDB. The StringDB is an incredibly useful database that is originally, I think, is mostly for protein-protein interaction network, but they also have gene annotation for over 2,000 organisms, and they have 1.4 billion interactions among different proteins. It also have gene ontology and CAC access. So one of the things that is possible with string database is not only the protein-protein interaction, it also enrichment analysis for bacterial genomes, which we don't have any right now. Okay, we have about 208 species, plant and animals, but we don't have any bacteria. But if you are working on bacteria, maybe string database is a very good resource. One of the things that I encourage you to try the StringDB is if you have a list of genes, you can paste it here, and the StringDB will realize the gene expression, will realize the protein reactions, also conduct enrichment analysis. One of the things, as you notice here, is this is very slow. It is very slow because we are converting your gene IDs into string IDs, and also we are pasting your data into the string database, your gene list data into string database, and to enable to access their data and annotation database. So just be careful, just be patient. Just be patient. If it's running, just let it run. If it doesn't run, that means the iDEP software cannot make a correct guess of the species with the StringDB. Then you need to search in this box. Only if it is not running. Okay, we are down here. So I get that started. So once this is finished, as you could see, it sends your gene IDs to over 1,000 of them, about 2,000 gene IDs to the StringDB, and it identifies the enriched gene ontology terms. Okay, so for upregulated genes, regulation of cell proliferation. For downregulated genes, we have again cell cycle. Okay, the p-value might slightly be different, but these results are very similar. Okay, to what we got from the annotation database we have. These results are entirely computed. The calculation and also the annotation database was on string database. As you notice, I also clicked here to get the next steps started. Before I move on, one thing that you can do here is also to switch to other databases for other annotation categories that you can run on the string database. One of the things that is not here is that you can see the enriched protein domains by using PFAM and interprom databases. And we also have CAC that's similar. One of the things what we are going to see next is the highly enriched is the protein-protein interaction networks among the differentially expressed genes. If you have too many differentially expressed genes, we're only going to use the first 100. Okay, and also we're going to do some. Looks like it's done. So as you can see, this is the protein-protein interaction networks among the upregulated genes. Not all of them, because we have something like 900 upregulated genes. We only use the top 100 upregulated genes and the protein-protein interaction network. So this network doesn't look very good, and a much nicer version is available here if you click here. But before we click, we also did some statistics here. So this is tells us, compared to random genes in the string database, your genes are enriched with protein-protein interactions. That means if it is enriched, that means your proteins are more likely to interact compared to random group of genes. That means your functionally is more coherent. Okay, so to get a much nicer and interactive network, what we are doing here, we can actually have a link, very customized link. If you click on this, will brings you to an interactive version of this network. But this network is StringDB. So this is your genes that iDEP pasted the 100 genes, the most operated genes on your network, on your differential experience to StringDB. So this is a customized network. What it shows the protein-protein interaction network, and it's inactive. You can move them, and also we have a lot of annotation database. For example, some of the larger proteins have their three-dimensional structure solved. So you can see their three-dimensional structure. You can see their protein domains. You can have annotation to the protein domains on the different database. You can probably click. You can also see the annotation of the gene, the full name of the genes, and their links to CAG, links to ensemble, and stuff, right? So this is the three-dimensional structure of different domains here. Okay, you can also click on each of the link and get the evidence behind the link. Which is the paper. StringDB have a lot of information. Some of the link is based on co-expression. Some of them are based on protein-protein interaction experiments. Some of them is inferred. So you want to make sure, and you can examine your data in here, and also get shots in here. So this is upregulated gene. Here is downregulated gene. It's the same thing. You got another graph. You got another network in here representing the downregulated genes. As you can see, the downregulated genes are more densely connected, more densely interacting. This is because, as we talked about, the downregulated genes are cell cycle related genes, and cell cycle related genes, as we know, highly interact with each other. Okay, the cyclins and a lot of cyclin genes, as you can see, are in here. CDC8, even just based on the name, cyclin A2, these genes. Okay, and the last network that is available to you is both up and down regulated genes, and also we send the full change values to StringDB. Here, the genes are highlighted by their. I think this is downregulated gene. This is upregulated gene. So, for example, here the KDR gene is very is significantly downregulated gene, downregulated, and this KRT34 is significantly upregulated. So the fold change. So on this plot you have fold change numbers. Okay. So by default we pick the top 100. So if you want, you can see, okay, I just need top 20 genes. Okay. If you increase this to 400 genes, and you might have tens of thousands of interactions among them. So the top genes was selected by fold change instead of p-value. Okay, as I told a lot of biologists, p-values measures the significance or the certainty about the differential gene expression, but the size of the effect, how much a gene is up or downregulated, is measured not by p-value but by the fold change. So if you want to do some PCR verification, or if you bet on some other downstream analysis, selecting your top genes by fold change is much better, I think, in my opinion, than the p-value. Okay, because it measures the size of the effect. So it's going to take a little bit, but it's going to be much better than the first time. The first time when you use this, it downloads the protein-protein interaction networks whole data for the species. Okay, so it does get better for. Okay, so it's done now, and for 20 genes, we have a nicely looking figure here. For upregulated genes, we have lots of chemokines, CXCL3. These are cytokines, and they all interact. This is again much nicer graphics is here. As you can see, so upregulated genes, for downregulated genes, again cell cycle genes, cytokines, cyclins, for example, here. Okay, if it's not connected, then it's all here, and both up and downregulated genes is here. So downregulated, upregulated KRT36. Okay. Okay, so KRT36 is here, and so that's the protein-protein interaction network. So again, the StringDB API really gives a lot of functionality that is not previously available in iDEP. So I'm so glad that I figured out. Not only does it support more enrichment analysis for bacteria and a lot of other species, more than 2,000 species. It also provides a access point for protein-protein interaction networks. Finally, so we're looking at Pathway Analysis tab. If you click on Pathway Analysis tab, we are conducting a pathway analysis using gene set enrichment analysis, which is the default. This is different from the differential gene expression analysis, where we use enrichment analysis on these selected subset of genes that are differentially expressed. For pathway analysis, we use the whole set of genes. We use all the genes for enrichment analysis for random statistics. Okay, so but here in iDEP, to simplify the workflow, we just use the fold change values. We don't do the permutation test, for example, permutating the samples for gene set enrichment analysis, and also we use a little bit faster version of FGSCA for using the gene set enrichment analysis. Again, here we see the cell cycle. Okay, for downregulated genes, we see cell cycle. Okay, for positive regulation of, so it's just a little bit different. Okay, chromatin segregation, spindle DNA replication. Okay, not even cell cycle. So it's a little bit different. But if we switch to a gate for package, so it will be slightly different type of result. We have cell cycle, and also we have DNA damage repair and chromosome segregation. So this actually supports the conclusion that these researchers observed, where these cells, when you knock down HOX-A1 gene, actually have cell cycle arrest in S phase. So it prevents it from transition from S to G1 phase, I think, if I'm not wrong on this. One thing that we we can do here is we can also switch to different databases. One of the very useful ones is KEGG. Okay, once you switch to KEGG, as you could see, not only we do the analysis. Obviously we do the cell cycle genes are downregulated, and cytokine is upregulated again, which confirms our enrichment analysis. We also have a pathway diagram that we grabbed from KEGG, and with gene expression overlaid on top of it. Okay, so by default the first one is here, and you can switch it to DNA replication, for example. We'll grab the second diagram. So be patient, because we are actually going to download this pathway diagram from KEGG and then overlay gene expression on top of it. Okay, so for example, we have a lysosome gene expression data. The pathway diagram, the gene expression data is overlaid on top of it. Okay, the lysosome also makes a little bit of a sense, because once we injected the siRNA into the cells trying to destroy it and move it for recycling. So that's where these lysosomes come in. Similarly, when we can download this data, we can have a tree of these pathways. Okay, for example, cell cycle genes and gene replications. They realize the redundancy and overlap. We can also have a network that shows how these sets overlap. Okay. One of the tricks again is that if you don't like how the networks is overlaid, you can zoom in and zoom out. Once you zoom in and zoom out, R will recalculate this data. You can download all the pathway data, not only the pathway name of the pathway, upregulated, downregulated, name of pathway again here, the number of genes, and also the genes in here. Okay. So iDEP is trying to make it open so that you can upload, you can change a lot of things. Sometimes, especially for KEGG pathway, for one single pathway, you can have some genes upregulated and some genes downregulated. So this is useful when. This is very useful. See, instead of using the fold change, we use the absolute value of the fold change. Okay, sometimes that is an option. You click on here. We are, obviously we're going to have a little bit different type of result. The top downregulated pathway now is a ribosome. Okay, so protein synthesis is suppressed when upon injection of siRNAs into the cell. Okay, so obviously you can specify what type of gene set you want to include for pathway analysis. If you have very small gene set, you might have some false positives, because the smaller gene set, the statistics might not be very reliable. But sometimes some gene sets is just small, so you want to be sure. Okay, the significant pathway significant cutoff, the number of pathways to show in here. Okay, so this is again, I have a warning here because there are so many combinations. There's 1, 2, 3, 4, 5 methods, and then here you have so many different pathway or 10 pathway databases. The combination, you have more than 40, 50 combinations, so it's very easy to generate false positives. And we know that there's so many literatures and so many publications that are not reproducible. It's very easy to rationalize any result, so you can make a story out of anything these days. Okay, don't be too excited. And one last thing before I move on to show you is that we have a PGSEA methods for analyzing gene expression. The PGSEA uses the package called PGSEA, but also we have some modifications here to it. So what it shows is, okay, for cell cycle gene is higher in control but lower in HOXA1. And by doing the in ANOVA analysis, we know that this type of distribution, is significant, is unlikely. And so cell cycle gene and lysosome is the second. So the ranking is not by p-value, it's by the absolute difference we saw. Okay, so the most important story here for this data is cell cycle, and secondly probably lysosome is increased. So these pixels are, you can think of them as average gene expression of cell cycle related genes in this sample. So this is 3 samples. Okay, so these cell cycle genes are highly expressed in here, low expressed in here. You might recall this is very similar to the principal component analysis result. Okay, there's 2 types of plot. Here you can plot as all samples, all groups, including also you can use just the comparison. Okay, so this is we have some. So the red means highly expressed, average expression is higher, and blue means lower. Okay, again you can switch here for pathway analysis. So this is the pathway analysis tool. One more thing before I go is that if you selected a non-CAG, if you don't select CAG, and then we are showing a heat map of the genes that belongs to each of the pathways below in here. So okay, one last thing that I'm going to show you with the pathway analysis is that you can, for each pathway, for CAG, we have the CAG diagram. If it is not CAG, we can also see the target genes and their expression profile in here. So, for example, here we can see here we are using microRNA target genes, all right, using PGSEA. So the top one is microRNA 193B, and we can see that all the the target genes are mostly lower here in the knockdown, and in the controls some of them are higher, but if we download this we can see that the data behind that heat map. One of the things that we can find is, for example, cyclin D1 gene that was target of microRNA 193B was downregulated here once on that OXY1 knockdown. This is actually, there's some paper about this showing that indeed 193B inhibits cell cycle, target cyclin B1 and inhibit proliferation, migration, invasion. Okay, all right. So there's a lot of information you can get from here, and that's the pathway part of it. And for the genome tab, in the genome tab, firstly we are visualizing gene expression on the chromosome 1 all the way chromosome X. Chromosome 1 probably have too few genes, was now shown here. And you can zoom in. So for example, for here we can see a group of the upregulated genes are red and downregulated genes are in blue. Okay. For example, here we can see some genes. We can zoom in, and to see what genes are here, we can move mouse over. For example, here is AREG gene that was highly upregulated, and in this neighbor, there's quite a few other genes, cytokines, I think. Okay, this is a cytokine neighborhood of genes. This is another chromosome 15 that we have another neighborhood that is downregulated, cyclin B2. You know, a lot of other genes in here, so you can zoom in here. So sometimes the change in gene expression are correlated in chromosome location. For example, you could have a cancer that have a chromosomal amplification of a region, so you have a gene upregulated in that particular region. So there is a R package called PrEDA, which runs some kind of regression analysis and trying to distinguish the signal from the noise. And its result looks like this. It's highlighted using FDR 0.01, it highlights these genomic loci that was are enriched in upregulated genes and enriched with downregulated genes. Okay, so if you see my archive paper on iDEP, so if we use some genes using Down syndrome data, we know Down syndrome have extra copy of chromosome 21. Using the expression data and with PrEDA, we can actually detect that the genes are significantly upregulated in chromosome 21. So you can download the loci. You can also see the genes. You can that is uploaded that are there. Okay, hopefully it'll show up pretty soon. If not, I'm going to skip that. Okay, so the last two tabs, we have bioclustering. Bioclustering is also only useful when you have a lot more data, a lot more samples. The idea is very simple. So if you have a gene expression like this, if you run there might be some genes, for example, gene 1 and gene 2 are correlated among subset of samples. For example, only correlated of first 4 samples, but not on the rest. So bioclustering classifies gene at the same time classify as samples. All right, so it also gives rise to gene clusters. So for example, here we have 7 clusters. Cluster 1 have 820 genes. So this is cluster 1 and the enrichment cell cycle related genes. These are the genes in the cluster. So cluster number 2 has 138 genes. Okay, it's also in this region, but hopefully I can find. Cluster number 4 has 11 genes. These 11 genes are correlated only on control 1, control 3, knockdown 1, knockdown 3. Okay, there's also quite a few methods that is available here, and so for more information, look at the help page on bioclustering. So the methods are listed here on the references, and you can download the biocluster. You can change for enrichment analysis as well. Okay. But again, this is used for when you have a lot more data. And the last one is co-expression networks. The co-expression network is mostly hype. Okay, but sometimes it's useful because the co-expression, the correlation of gene expression is best summarized by this clustering heat maps. But if you have to look at co-expression network, and this is what it looks like, what it does is it takes your expression data, takes a normalized expression data, and runs the WGCNA package, and we have the gene dendrogram here with the colors, different clusters, and then shows up the co-expressed genes on the top 10 genes. Obviously, you can change this into top 20, and you can change it to the layout and cutoff. Okay, and you can download it. And then we have also the enrichment analysis for the top 10 genes. As you could see, there's cycling related genes and SOX4 and stuff. A lot of probably cell cycle related genes, as we can see from here. So again, this is the quick summary of the iDEP app. If you have questions, feel free to email us. And if you are having trouble formatting your data to upload to this website, send us your data, or first few column rows of your data. We can help you set it up. And also there's a user group on Google, and also there's a Twitter account. The source code is available here on GitHub, where you can see what's really goes on behind the scenes. And also we trying to generate the environment there you're using, what version of the R package you use, and for your record you should be able for reproducibility issues, and then I have a change log in here what I did update. And this is I wrote most of the R code. My student Kevin Song helped setting up the server and help scale up the server and maintain the server, and he figured out a ton of stuff that is very important for the server. Again, let us know what you think, and thank you for your attention. So there's a bioarchive paper on this if you have more questions. And I'm also very active on Twitter, so tweet me or send me email. Thank you very much.